In section 3, we'll be looking at how your skeletal muscle cells or fibers transmit an electrical signal to get your muscles to contract. Now, that electrical signal is what's called an action potential. And so there's a lot of terminology in this section, so we need to really pay attention to some of the diagrams and things to try to help explain the process and to learn the vocabulary, the terminology. So on the picture here, I have an example of a cell membrane. The cell membrane is, could be a muscle cell, skeletal muscle cell membrane. It's a lipid bilayer. It has many different openings here. These are all different ion channels, which we'll talk about in a few minutes. And then you see molecules on both sides of this membrane. So on the inside of the membrane, all the little purple dots here, that represents potassium ions. All the pink dots represent sodium ions. And then you have some protein molecules that are inside the cell, inside the membrane, and they're representing negative charges. And so in general, what you'll find is at rest, at what's called the resting membrane potential. This is when a muscle cell is not contracting. It doesn't have an electrical signal. So at rest, it's usually more negative on the inside of the cell and more positive ions on the outside of the cell. That's why I have the positive and negative here. That creates what's called the resting membrane potential. That type of situation is set up using a sodium and potassium pump. The sodium-potassium pump helps to create a concentration gradient with the sodium and the potassium ions. Now you have to have this type of setup, this resting membrane potential, to create an action potential, which we'll see in a little bit is a reversal of the charges. So for an action potential to be created, you'll have to reverse the charges, and you'll have to take the sodium ions and move them inside the cell, and that creates a more positive environment on the inside of the cell. If you could measure inside, if you had a probe that could actually go inside of the cell and actually measure this, you would see that you actually have a negative charge on the inside of the cell. So again, you have a diagram here that represents the plasma membrane, the cell membrane. They're looking at a nerve cell here, but it would be the same for a muscle cell. You put in a probe and look inside of the cytoplasm, and you would find that it's relatively negative on the inside and it's going to be positive on the outside. They estimate here for this particular cell that the resting membrane potential would be a range from around -70 millivolts to -90 millivolts, and that's inside the cell. Now, the ion channels that I mentioned just earlier, these allow for ions to move back and forth across the membrane. And so there are openings in the cell membrane. They're usually proteins that kind of form a tunnel inside that cell membrane. And there's different types of ion channels. So for example, you have leak channels. These are just channels that are usually always open. There's no gate. That means ions can move back and forth, especially like potassium usually has a leak ion channel for potassium ions. That allows potassium to move back and forth, really based on diffusion. And so the higher the concentration, if you have a concentration gradient, you have higher potassium on one side of the membrane, that allows it to kind of push through the membrane, through the ion channel to the other side of the membrane. Other types of channels are gated channels. They have a gate type mechanism there that has to open in order to allow the ions to transport across. And so a ligand gated channel, ligand is just another name for a chemical or molecule. And so ligand gated channel only opens whenever that molecule or that ligand is in place. It has to bind to that protein channel. When it's bound there, then that allows the gate to open, that allows the ion to pass through. Now, usually neurotransmitters work as ligands. They work as molecules that help to open these channels. And so a good example, which we'll see this one a little later, but acetylcholine is a type of neurotransmitter, and when it binds to a sodium-gated channel, a sodium ligand-gated channel, it causes a sodium gate to open, and that allows sodium ions to move into the cell. You can also have voltage-gated channels. They work in a similar way, but instead of responding to a molecule or a ligand, they respond to a change in voltage, an electrical signal, and that causes the gate to open. We'll talk about later an example where you have a calcium channel that's a voltage-gated calcium channel that responds to a small change in voltage to open that channel. So I mentioned briefly earlier that an action potential, that's the electrical signal, is really a reversal of the resting membrane potential. Remember that resting membrane potential means that you have more negative ions on the inside, more positive ions on the outside. So to get an action potential, you have to reverse the charges. Well, the question is, how do you reverse the charges? To do that, you have to open up these gates, the ones that are closed, the ones that are gated. You have to open those up to allow the ions to move different places. And so it's kind of hard to see the diagrams here, but make sure you go back and look at some of the diagrams. We'll kind of briefly look at a few things. Here you have that same membrane. You got the channels here. These happen to be, both of these are gated channels. One's a sodium channel, one's a potassium channel. You've got to set it up at rest, so it's more negative here on the inside and more positive on the outside. Notice on the graph, they've basically graphed what's taking place inside of the cell. So the graphs here, which we'll talk about, is what's going on inside the cell. So right now, this is time on the x-axis, voltage, millivolts, membrane potential on the y-axis. They measure it. If you were to measure inside this cytoplasm of the cell, it would be at rest, and it's about a minus 90 millivolts. Then to have an action potential, you reverse the charges. To do that, the sodium gate must open. There's got to be a signal to the sodium gate. Usually this is a voltage-gated sodium channel. So a change in voltage causes this sodium gate to open, and all the sodium, or not all of it, but a lot of the sodium here wants to go based on diffusion from a high concentration to a low concentration inside the cell. Then notice on the graph, it causes this line here to sharply increase very, very quickly. And when we're talking about less than a millisecond, you've increased this line. In other words, you've gone very positive, right? This is taking a picture of what's going on inside the cell. That inside the cell just became very positive now. It went from minus 90 to almost a positive 30 in less than a millisecond. That's how quickly this is taking place. This is what's called depolarization. The cell membrane has become depolarized, or the cell has become depolarized, just because the sodium ions are now flooding inside and getting into the cytoplasm. Then the second thing that's going to happen is a repolarization stage. That sodium gate is going to close, and eventually a potassium gate is going to open. And then you also have leak potassium channels that are always open, and that allows for the positive ions that were flooded in to go back through. And again, it's based on concentration gradient. So you have potassium ions to flood back through, and they exit the cell, and that causes a sharp decrease in polarity or drop in the charge here. So now you're going from somewhere around a positive 30 all the way back down to minus 90. And so that's called a repolarization stage. And so again, I have these defined for you, and another picture that I found that kind of shows this a little clearer. Depolarization inside the plasma membrane, inside the cell, becomes less negative, that's because it's becoming positive. And so just however you want to word that, either way is correct. It's less negative or it's positive. It moves toward the positive portion here. That's depolarization. Repolarization is returning back to the resting membrane potential. They show it somewhere around minus 85. And again, I have some other graphs here that kind of show you what's going on. This is at rest here. You have sodium out here, a lot of potassium inside, more negative on the inside, more positive on the outside. Then you see depolarization is when the sodium gates open and the sodium floods into the cell. That creates more positive on the inside of the cell, more negative outside the cell. And then repolarization is the opposite of that. The sodium gates close and potassium gates open, or maybe you have a leak potassium gate or leak potassium channel that's always open, and now that allows the potassium that has built up inside the cell to now go outside. That's repolarization. So in this process, by reversing these charges, you've created what's called an action potential. You've created an electrical signal. Now that's going to eventually cause some type of reaction in your muscles. For example, that will eventually lead to a muscle contraction. And so one thing we'll talk about here is what's called the action potential propagation. That's just how that action potential is moved throughout the cell membrane. It propagates, kind of like a wave-like action. And so, for example, you create an action potential in one section of the cell membrane, one section here, not the whole cell. The whole cell doesn't, or the whole muscle fiber. Remember how long the muscle fiber is. It doesn't get the action potential all at the same time. It's very, very quick, happens very quickly, but it's not all at the same time. And so one section becomes depolarized, and then that stimulates the next section to become depolarized, and that stimulates the next section to become depolarized. And the process continues down the length of that muscle cell. Now, let's kind of combine some of the terminology that we've seen before and this information about action potentials and see if we can learn how that muscle fibers, muscle cells go through the process of contraction and relaxation. That's section 4. A few other terminologies we need to look at here. For this to take place, you have to have to get the signal from the nervous system to a muscle cell. So here we have a muscle cell. Here we have the end of a neuron, the axon, that's coming up in contact with a muscle fiber. So it's going to be sending a signal, an electrical signal, an action potential down the length of the axon. And it's going to travel the same way with the depolarization, repolarization, the propagation, all that's going to travel the same way down the length of a nerve cell as it will down the length of a muscle fiber. So that action potential has to travel down, and then at the end of the axon, you have it's what's called the terminal. The end of the axon is a terminal, and so here you have what's called the presynaptic terminal, which is the axon, the end of the axon. The postsynaptic membrane, that's going to be what's after the synapse, which is a junction or a space area. And so in this case, that postsynaptic membrane is going to be the membrane for my muscle cell. Right? So presynaptic, before I get to this junction, and then postsynaptic after I cross the junction. So the junction is called the synapse. The junction is a space here. Sometimes you see the space called a cleft. And if you look at the enlarged area here, here's the end of the axon. This is, again, called the presynaptic terminal. Here is the space here. That's called the cleft. All of this is what's referred to as the synapse. It's the whole junction. It's where an axon meets a nerve. An axon meets a muscle cell. It could be a axon, a nerve cell meets a gland cell, or a skeletal muscle cell or a smooth muscle cell. Whatever the junction is, it's called a synapse. In our example here, we're dealing with a neuron axon meeting a muscle cell. Right? So inside of the presynaptic terminal, in this case, inside of this axon, you have little storage vesicles. We'll talk about these. These are storing neurotransmitters inside, chemicals inside of this axon terminal. One other term here to mention is sarcolemma, and that is just a name for the membrane for the muscle cell. Instead of calling it a plasma membrane, they call it a sarcolemma. Also notice the word sarcoplasm is there, and that's just the cytoplasm of the muscle cell. Again, synaptic vesicles, they store up chemicals, and those chemicals are called neurotransmitters, and you can kind of see the definition there. These are chemicals or substances that they get released from the presynaptic membrane. And we'll talk about this process in just a minute. They diffuse across, they go into that cleft area of the synapse, and they can stimulate or inhibit an action potential on the next membrane. So they're in charge of basically bridging the gap from the axon here to the cell membrane here. So in this case, I've got a nerve cell and I've got a muscle cell, and I've got a space there. Well, you've got to get an action potential that's coming down the length of this neuron. You've got to get a new action potential over to the muscle cell. And to do that, you use a chemical type of a junction here, which is called a chemical synapse, and you use neurotransmitters, and the main one we'll look at is acetylcholine. and that helps to create a new action potential on the next membrane. To help stop the process, because eventually you've got to re-establish your resting membrane potential, and so you've got to get rid of this acetylcholine from the storage vesicles, and we'll kind of see that process in a little bit. You have an enzyme. It's a very long word, but it's an enzyme. It ends in ASE. That's important if you don't know that. ASE refers to an enzyme here called acetylcholinesterase. This enzyme helps to break down, when you're done with the acetylcholine, you need to get rid of it. So this acetylcholinesterase breaks down the neurotransmitter acetylcholine. So this diagram tries to help put some of these things together. It's a big process. And again, we're trying to send a signal, an action potential, from our nerve cell down to our muscle cell to get our muscles to contract. And so this diagram 10:15 is trying to explain some of that. Here's your action potential coming down the length, the axon, coming down the neuron's axon here. As it comes down, it hits it stimulates a calcium channel. This is a voltage-gated calcium channel. It stimulates that calcium channel to open, and so calcium ions can now flood inside of this axon terminal. Well, calcium causes that acetylcholine that we just mentioned. That acetylcholine is stored inside of these storage vesicles. And so it turns out that calcium, when it binds to these storage vesicles, releases the acetylcholine. Now the kind of triggers the acetylcholine to be released from the vesicles, and that enters into the synaptic cleft. The acetylcholine is going to work like a ligand, or it is working as a ligand, because it binds to the sodium-gated channels, to the ligand-gated sodium channels on the next membrane. And when that ligand binds, guess what? It opens the gate here, and that causes sodium ions to go inside of the muscle cell. What you've done now is created a new action potential, because you've just reversed the charges. This was a membrane at rest. A sarcolemma was a membrane at rest here, a cell at rest. It had more negative on the inside, more positive on the outside. But because the acetylcholine binds to the sodium channels here, it opens them up. It reverses the charges. You have depolarization, and now you've created a new action potential. That new action potential is now going to propagate down the length of the muscle cell in both directions. Here's another diagram. This is just from a different textbook. It kind of says the same thing, so look over this. You can follow the pathway. I'm not going to review it again, but it kind of follows the same pathway. This is also in your notes. Just kind of go through this again and go step by step, kind of learn what's taking place to get the signal from the axon here, axon terminal, to jump across to go to the membrane of the muscle cell. Notice also it lists the acetylcholinesterase here. That helps to break down the acetylcholine. That helps to close off your sodium channels here. So as long as the acetylcholine is binding to the sodium channels, then the sodium gates are going to be open. And so you got to get rid of that acetylcholine to close the sodium channels, to reestablish your resting membrane potential, to allow your membrane to become repolarized. And so that's why you have to have the enzyme acetylcholinesterase, and that clears out the acetylcholine that's in the synaptic cleft area. Now, to see how this actually causes a muscle fiber contraction, we need to look at a few more terms and kind of put everything together. This involves all these different parts we've talked about. Sarcolemma. Now, the sarcolemma, again, is the membrane. So you got an action potential you've just transferred to the membrane. And so here's our muscle fiber here. And here's our little dotted lines represent the action potential. And so it's going down the length of the membrane. You have something called T-tubules or transverse tubules. That's just an inward projection of the membrane. So it's best seen here in this lower diagram. That allows the action potential not just to go along the outside of the cell, but actually allows the action potential to go inside the cell as well. And it needs to do that because it needs to release a storage of calcium that is stored up inside the cell. So inside of the muscle cell, you have something called sarcoplasmic reticulum. That looks very familiar to something called endoplasmic reticulum. It's a form of the endoplasmic reticulum, but in this case, its job is to store calcium. It stores up calcium. And of course, we're going to talk about the troponin and the trypamyosin here as well. The end of this area is called a cisterna, or cisternae is plural, and that's just kind of where a lot of this calcium is being stored at the ends of this. At the end of the sarcoplasmic reticulum, you would find the terminal cisternae. So let's see if we can watch the whole process now. We got our action potential. We got it transferred from the axon over to our muscle cell. It's going down the length of the muscle fiber. It goes inside the cell where it opens up a calcium reserve inside of the sarcoplasmic reticulum. So now you have calcium on the inside of the cell. Calcium's job is to bind to that protein we mentioned earlier called troponin. Remember we said that troponin has several different binding sites. It binds to calcium, it binds to the trypamyosin, it binds to the actin myofilaments. Well, now you have the calcium come in, it binds to the troponin. When calcium binds to troponin, it basically causes a shift in that molecule. So now troponin kind of shifts around. When it shifts around, it moves that trypamyosin thread-like protein out of the way of the binding site. So now calcium binds, it shifts to troponin. See, it's kind of changed shape a little bit. And when it does, it kind of pulls away that string-like trypamyosin. When that happens, it exposes an active site on the actin molecule, and it allows the myosin protein, or the myosin myofilament, to bind to the actin. That's your cross bridge formation. You've got to have the calcium there to get the cross bridge formation to take place. Figure 10.18 from your textbook kind of goes through this process, and it kind of shows you the process of what's called cross bridge cycling. And it kind of shows you, it's kind of hard to see here in this diagram. So take a look at this in your textbook or in the PowerPoint notes, and you can see, once the calcium binds, it shifts that molecule out of the way. You can form a cross bridge here. You've got the hinge action. Once the cross bridge forms, you've got energy molecule that's here. The energy molecule causes the head of that myosin molecule to shift or to hinge. When that does, it's pulling that actin further down, and that creates that contracting motion of the sarcomere. So you're shortening the sarcomere when this is taking place. Relaxation means you've got to get rid of the cross bridge. To get a muscle to relax, you've got to get rid of the cross bridge formation. And so that takes energy as well. You have to dislodge or disconnect the head of the myosin molecule from the actin, and then you have to move the calcium back into the storage container that it was in. And so you're kind of following some steps here. Here's the axon terminal. There's our little synaptic cleft that we talked about, and the synapse that we talked about. You got to get rid of the acetylcholine, so that's the job of acetylcholinesterase. Then you've got to pump back in the calcium, put it back in the storage area, so that you get rid of the calcium so that your troponin can go back to its normal position, so the trypamyosin can go back to its normal position. And that means that it covers up those active sites on the molecule, and that means your cross bridge will basically dislodge. Your myosin head will dislodge from the actin.